first name. You guys have the same first name. No last name. All right, less than a minute to go. Let's call out the names who are not here to be sure. Got, so if you hear your name, obviously correct me. Roselle, Sims, Valdez, Page, Carr.
You said, you telling
No, Carr.
oh, what are you trying to be short for?
Oh, no. Do your coaches ever do that?
No, I never got that.
My name was always abbreviated by coaches and drill instructors. No one ever called me D. Ellis, just D. Anyway, Smith. Scout student's not here. Stanley. Strickland's here. What's that? Okay. And Vega. All right. So anybody I called is indeed here. It's not. All right. Let's get started. Yeah, it's 8 o'clock. We'll get started. So I still plan to release the retake for the first exam for those of you who want to take it. But also,
When is that due?
whenever, there's no due date. It's a great question. So once I release the first exam's retake, you can take it on the last day of the semester if I want, if you want. Matter of fact, it might help you to do it the weekend before the final. It'd be a good way to say for the final, because part of the first exam is what I'm going to use to write the final. I'm literally going to take the questions off the first exam, the second exam, and the third exam, verbatim, and put them on the final. So it might be a good way for you to study. It's a great question. No due date once that retake is posted. I will say, since we had this last conversation, like I said, on Friday, I was so disappointed in some of those emails I got and some of those people that missed the exams. That was rough. And like I said, because of that and being sick, I kind of stopped doing the grading for a bit, but I finished it now. And I've got to say, for those of you who took it, it's really good. The lowest grade was a 70. So of the people who took it, no one failed it, no one even got a D. So I'm really excited. Everybody did very good, very well, everybody who took it. And again, for those of you who haven't taken it yet, I'm going to post that retake soon. So technically, this will be your retake, even though it'll be your first time. So whatever you get, it's just going to be average sum of your first grade, which was 0. So any questions about the first exam? Again, great job. Especially considering a lot of people didn't do the study guide, so I'm pretty impressed. Though I still recommend doing study guides. Another thing I'll remind you is lab is today at noon. Don't be late. Make sure you're wearing long pants and closed-toe shoes. Please, please, please read ahead. Because, like I keep saying, sometimes, and I did this as a student too, you get so caught up in the details, step 1, step 2, and you're just following things and trying to make sure you don't miss anything, that you miss the big picture. So if you're really familiar with what you're getting into before you get into it, hopefully it'll help you see the big picture. And also, for all of us, the more you know what you're doing, the quicker we're out of there, right? I know no one wants to stay until 1:50, me included, so, be prepared. What else? I guess that's it. Any questions? All right, let's jump back into chapter 5 then. We are talking about basically an overview of energy, basically, right? And how energy works as far as the cell is concerned. So first we talked about some basic energy concepts. I'm not going to go over everything, but some big stuff you definitely need to know is that the 2 basic types of energy are kinetic and potential. Everything else we talk about falls under those categories. For example, the big one we're going to talk about for this chapter and the next 2 chapters is chemical energy. And chemical energy is a type of potential energy, because remember, potential energy can be something due to location, like this thing has more potential energy than this. Well, if you imagine they were the same thing, because it's higher. But more importantly, for us, and anyway, potential energy also includes stuff by its structure, right? However much energy it took to build something, that's how much energy could be released when you break that thing. That's the most basic way of putting it. So make sure you understand that. Make sure you understand the conservation of energy. So energy can't be created or destroyed. It can only be converted from one form to another. Every time you do that, every time you convert energy from one form to another, you do lose some of it as heat. Those are the most basic concepts from that main bullet point. Then we started talking about energy transformations, ATP, and cellular work. If anything, the most important thing you need to know from that bullet point is what kind of energy do cells use directly? ATP. That will be a test question. What kind of energy do cells use directly to manage with ATP? And right now, that's an easy thing to remember. So far, that's all we talked about. By the time the exam comes, though, it's going to be a little bit more confusing. Because in the next chapter, we're going to talk about how we make ATP. And how do we make ATP? We basically burn glucose. So that will be one of the options in the exam. So you might think, all right, cells use glucose as energy. But not directly, right? First, they've got to break down the glucose to make ATP. Anyway, now we finally got into enzymes. And let me back up a little bit. Mind you what we talked about with the enzymes. Get it pulled up here. There we go. Yeah, so far the only thing I'll introduce you to is this word called metabolism, which is basically all the chemical reactions that are happening in your body. And then your book points out that all these different chemical reactions that are happening in your body, each one of them requires a specific enzyme, basically. And then we talk about what an enzyme is. And this is very important. You need to know this. Enzymes are proteins. That's the most important thing you need to know. Well, one of the most important things you need to know about them. They are proteins, right? So they're not carbohydrates, they're not nucleic acids, they're not steroids, they're proteins. What do? According to your book, they speed up chemical reactions. And then, like I said on Monday, not only do they speed them up, a lot of times they just make chemical reactions happen that normally just wouldn't happen without them. So, very important. And again, they are not consumed. And I'm going to come back to that concept here in a second. And that's where we left off. We are now caught up. Let's talk about activation energy. Activation energy is the energy invested to start a reaction. So what I didn't tell you is that sometimes chemical reactions give off energy. Sometimes chemical reactions take up energy. But either way, even the ones that give off energy, they still require a little bit of energy to get started. And that is called activation energy. Technically, what that means is this is the energy that's needed to activate the reactants. As far as I'm concerned, you don't necessarily need to know that. Not for the exam anyway, that's just kind of for your own knowledge. It activates the reactants. And to remind you, for chemical equations in this class, we have reactants on the left, and you have the arrow pointing, and then whatever's on the right are the products, right? So again, the activation energy is what's required to activate those reactants. And then once you do that, that triggers the chemical reaction, which is basically like saying you have all these chemicals in your body, right, ready to do a chemical reaction. But luckily for you, they're just not constantly happening. Life wouldn't work that way. But when they do need to happen, that's when we have the activation energy gets lowered by the enzymes, and then these things can happen. So again, enzymes are proteins. They are not used up in chemical reactions. They speed up chemical reactions, or sometimes start them when they wouldn't start at all. Here's the new information. How do they work? They reduce the amount of energy required to break the bonds of the reactant molecules. In other words, put it better. I'm going to just put it this way. I'll put a line through this. If you're taking notes, the better way to remember what enzymes are, you know, this is kind of saying the same thing. Enzymes reduce activation energy. It's that simple. So again, to start a chemical reaction, you have to put some energy into it. All enzymes do is lower that amount of energy that's required. Speaking of which, the first word for attendance will be energy. So, how many of you have ever shot multiple weapons? Whether it's, or let me rephrase that. Multiple weapons that have triggers on them. Anybody? Okay, just one. So, you might be the only one who gets this, or maybe somebody online. Sometimes you shoot like a gun, for example, and the trigger's kind of hard to pull. Sometimes it's really hard, sometimes it's a little hard, sometimes it's like a little feather trigger. Like you got to really carefully, barely pull it and you shoot it, right? So that's basically what an enzyme is. So we've already used this analogy of the crossbow, right? You pull the string back, and once it clicks, all that energy is ready to be released, right? But it's not going to be released until you pull the trigger. That's the activation energy, right? There's all that energy ready to go, but you still have to pull the trigger. Now, obviously, however much energy you're putting into pulling the trigger is not nearly as much as energy is coming out, you know, from the dangerous end of it, right? Otherwise, why would you pull the trigger? Why would you shoot somebody if you just do that to them, right? But that's not how it works. You need the energy that gets released when you pull the trigger. So what an enzyme does, basically, if you've ever shot other guns, it's like you have a really hard to pull trigger, and this one just makes it really easy. Like you barely touch it, now it shoots. Does that make sense, at least to you? Yeah, so again, enzymes lower the activation energy. I'm gonna show you some visualizations of what I'm talking about. This is really hard to visualize without really getting into the physics of chemical reactions. But basically, so this molecule has a lot of potential energy. That's what it means when it's up here, right? There's a lot of potential energy in that molecule because of the bonds that keep it together, right? There's a lot of energy put in when that molecule was built. And then once it's broken, again, it's at a lower energy at that point because it's released its potential energy, right? But the problem is, to get from here to here, you have to have this activation energy. And again, all the enzyme does is lower that activation energy. So you're still going from this amount of potential energy to this amount of potential energy, right? That part hasn't changed. It's still the same amount. You're still giving off the same amount of energy. It's just you're making it easier to start that reaction. Make sense? All right. The next part is the process of science, as every chapter has, and we will always skip it. So I'm going to skip it right now. Again, as always, I still recommend reading them. There will be questions about it on the study guide, I think, but not on the exam. And of course, as always, if you have questions, let me know. All right, let's talk about enzyme activity. So basically, you know what they do, right? You know that enzymes speed up chemical reactions or allow them to happen when they normally wouldn't happen. How do they do that? They do that by lowering the activation energy. Let's dive in a little bit deeper. First of all, you need to know that enzymes are very selective. That will probably be a test question. Trying to think of all the different ways to say that. They're very selective, they're not very broad, they're very specific. So again, like I said, there's maybe like an enzyme, for example, that breaks up lactose into its two parts, which is galactose and glucose. And that enzyme that does that job, it probably does not do any other thing, right? It's very specific to the chemical reactions that it does. So that's the first thing you should know. Next thing, enzymes recognize something called a substrate, and that is the reactant molecule. So whatever molecule is doing a job on, that's called the substrate. You need to know what that word is. Even if I don't ask it on the exam, I'm going to use it a lot. Throughout the semester, well, a good chunk of the semester. Because a lot of the things we talk about involve enzymes. So yeah, know that the substrate is the reactant molecule. Here's a little picture. Here's an enzyme. Here's the substrate. Here's the molecule that it's going to do its job on, so to speak. Where does the substrate fit into the enzyme? In a place called the active site. The active site has a shape and chemistry that fits the substrate. And it's very easy to visualize right here how the shape fits the substrate, right? In this example, you know, the active site's kind of curved right here and then sharp right there. Just like the substrate. It's curved right here, sharp right there. It's easy to visualize the shape part of it. What's a little bit harder to visualize is the chemistry. So yeah, the chemistry fits too. So for example, if this was, I don't know, a positively charged molecule, then obviously this part would be negatively charged because positive and negative go together. Make sense? Or if this part was polar, then this part would also be polar because remember, polar molecules go together, right? Non-polar molecules go together. Hydrophobic, hydrophilic, all that stuff. That's what we mean by the chemistry fits too. So yes, obviously the shape fits as we can see, but also the chemistry will fit too. So now you know what a substrate is and what an active site is. Now what do we call it when the substrate actually comes into the enzyme? It is called an induced fit. Any idea why I have a picture of people hugging? Because if you don't think about it, that makes no sense. But if you think about it, it might make sense. In the context of what we're talking about. Any guess why? All right, well, let me ask you this. I'm not going to demonstrate. Probably get sued or something, or fired. But if I were to ask somebody to come up here and give me a hug, well, first of all, a hug looks like this. Wouldn't you agree? Something like this, that your arms wrapped around somebody, right? But when you start a hug, do you start a hug like this? No, you start your hug like this, right? Arms wide open, and they come in, and then you squeeze, right? That's what an induced fit is. So the enzyme is kind of wide open like that, waiting for the substrate to come in, and then once the substrate comes in, kind of closes up around it. And that's what an induced fit is. So these are all words you need to know, maybe because they'll be on the exam, but mostly because I'm going to be using those words. So again, the substrate is the molecule that the enzyme is working on. The substrate attaches to the enzyme at a place called the active site. That's the noun, that's the place, right? That's the noun. The verb of that thing happening, that's the induced fit. So any questions about that? All right, pretty simple stuff. I'm trying not to go too fast. So after the product is released, the enzyme then is able to accept another substrate molecule, which goes back to one of those first things I told you about when we talked about enzymes, which is very important. Enzymes are not used up in chemical reactions. So again, this enzyme, it works on this substrate. The substrate attaches itself. It does its job of breaking that down, probably a hydrolysis reaction. And then the products leave, and then look, the enzyme is ready to go again. So the enzyme is not used up in a chemical reaction. And that is a key characteristic of enzymes. So there will be some questions on the exam. You're going to see a chemical equation that you've never seen before. Oops. And I'll go ahead and give you a hint. It says x plus y plus z gives you, I don't know, a plus b plus z. If I give you that equation, and I will give you some equation, except it won't be letters, it'll be some chemical you've never heard of, probably some molecule you've never heard of. If you see something on both the left side of the reaction and the right side of the reaction, that is the enzyme. So I'm going to give you an equation you've never seen before, and I'm going to say which one of these things is the enzyme. And again, if it's on the left side of the reaction and the right side, that means it wasn't used up, right? That means that is your enzyme. The other thing you need to know is that they always end on the word ace, with the suffix ace. For example, what I say for lactase, right? So lactase, right? It would be on the left side of the enzyme, left side of the reaction and the right side. So if you, again, if I show you a chemical reaction you've never seen before, if it's on both sides of the reaction, then you know it's an enzyme. Also, if it ends in ace, it's the enzyme. Nothing else will end in ASE, right? So those are two ways you tell if it's an enzyme.
And always, it'll always end in ace.
In this class, yes.
Okay.
And almost always in, broader conversations. But yeah, as far as we're concerned, they always end in ace. So those are your ways of knowing. And again, you will get that test question. And then the follow-up test question will be, after I ask you what the enzyme is, it'll say, okay, which one is the substrate? And again, well, not again, sorry, I'm going to tell you this now. Many enzymes are named for the substrates. And then in this class, all the enzymes are named for the substrates. So again, if you see this reaction, and instead of a Z, it's called lactase. I'm just giving that one example. Lactase, and you've identified that. There you go. Lactase is definitely the enzyme. And then one of these other things is called lactose, and ask you what's the substrate. The answer is lactose, because again, the enzymes are named after their substrate. Does that make sense? First, you identify the enzyme. It's going to be the thing that ends with ace, and it's going to be on both sides of the equation. And then just look for the thing that sounds like the enzyme. So again, in this example, lactose would be the substrate for lactase. Excuse me. Any questions about that? All right. And the next slide is going to be a picture of everything I just told you about. And it actually is the enzyme lactase. So even though I was just using that as an example, that's the one we'll look at. So there's the enzyme lactase. It's ready to do its job. Its active site is wide open. What is its substrate? I could ask you that, even though I just told you. But if I just gave you this picture and showed you some enzyme you've never heard of, you should be able to guess what its substrate is, because again, the enzymes are named after their substrates. So lactase meets with lactose because that's its substrate. It comes in to the active site and then has the induced fit. right? And the enzyme does its job, which in this case is to break up that lactose into its two parts, right? Well, we need to get, oh yeah, I mentioned this earlier. Though we already passed this on the first chapter, our first exam. This is a hydrolysis reaction, right? Because water is coming into the reaction. Anyway, the enzyme does its job. The lactase is broken up into its two parts, glucose and galactose. They leave the enzyme, and now the enzyme is ready to do its job. So obviously, this is just an example. I'm not going to ask you, what is the, you don't need to memorize the substrate for lactase, or you don't need to memorize that the two parts of lactose are glucose and galactose. This is just an example. Just showing you generally how an enzyme works. And when we study it this way, it's actually pretty simple, right? Substrate comes in, enzyme does its job, products leave, the cycle continues. So, any questions about that? All right, the next thing we're going to talk about is enzyme inhibitors. First of all, before I even give you this information, let's talk about why these things are needed. You can almost think of your cells as little factories, constantly building stuff, right? And they're only building stuff with the stuff you give it, right? So the things you're breathing in, the things you're eating, the things you're drinking, that's what it's working with, right? And there's limited supplies of things. So it can't just constantly build stuff. It only builds what's needed at that time. So how many of you guys remember fidget spinners? You guys might be too young. Now you should be able to remember fidget spinners. Remember those things? I mean, they're still around, but remember at one point they were really, really popular, like ridiculously popular. So imagine you're a toy factory, and then it's that time, I forgot when that was, maybe 2010-ish, I don't know. Imagine though, you own a toy factory, and then it's the fidget spinner craze. You're gonna put all your resources, or at least a lot into fidget spinners, right? I'm like, all right, quit building other stuff. We need more people, more machines, more resources to build these fidget spinners, because they're selling like hotcakes. You do that, right? And then all of a sudden the craze goes away, so now you go back to making other stuff instead. That's sort of like what an enzyme inhibitor is. Because enzymes, again, they don't need to constantly be making whatever it is that they make. Sometimes they need to make it, sometimes they don't. So your body needs a way, your cells need a way to turn them on and turn them off. So what we're talking about here is how to turn them off. And that is an enzyme inhibitor. Basically temporarily stops enzymes from doing the job. Or as your book says, enzyme inhibitors inhibit metabolic reaction. But again, that's just a fancier way of saying they stop the enzyme from doing what they're doing. So if you're not in a biology class and someone says, be careful not to drink too much because alcohol will lower your inhibitions. What does that mean when they say inhibitions? In that context. Does that mean that alcohol lowers your body temperature, or lowers your stance, or lowers your voice? What does that mean? Lowers your inhibitions. When I say alcohol lowers your inhibitions. No guesses? You guys have to know this. This is actually good knowledge for college students. No one knows? All right, so inhibitions are the things that keep you from acting a fool, basically, right? So saying something you shouldn't, doing something you shouldn't, and alcohol will lower your inhibitions, meaning alcohol will keep you or prevent you from stopping that, right? So just let you run free, and that's not a good thing, right? So anyway, I guess if you guys didn't know that word, that won't help you. But I was getting at, that's what enzyme inhibitors do, right? They keep enzymes from doing what they're supposed to do. Anyway, how do they do that? They bind to the enzyme, and when that happens, the binding disrupts the function. I guess the most important thing, as far as the exam is concerned right there, is that the enzyme inhibitors bind to the enzyme. That's how they do their job. There's two different types. The first one we're going to talk about is this one, the substrate imposter. Substrate imposters plug up the active site. And I know this isn't just a meme, but it's a great example. So here you can see this enzyme, this yellow enzyme, right? That's its substrate, this blue thing. And you can see why, right? That blue thing looks like it would fit in there just perfectly because that's the substrate. That makes sense. But here we have this imposter. Now it doesn't fit just like that, but it can plug up at least that one part, right? And because it's plugging up that one part, the substrate can't come in, the enzyme can't do its job. And that is what a substrate imposter is. It plugs up the active site. And you need to know that for the exam too. So again, to recap, enzyme inhibitors keep enzymes from doing their job. How do they do that? They bind to the enzyme. There's two different types that bind to the enzyme. First one is this one here, substrate imposter. It binds to the active site. It literally blocks business end, if you will. Any questions so far? Okay, let's talk about the other type. And there's words for this. Your book doesn't give them, so I'm not going to require you to know them. But if you want to look it up for independent work, you could. What are the actual names of these two different types of enzyme inhibitors? Anyway, other than the substrate imposters, we have the ones that bind at a site other than the active site. So here's a good example. Here's this enzyme. You know, its substrate kind of looks like a pumpkin teeth, I guess, if you will, two little squares, right? So that's the substrate, it fits there. But this one doesn't bind there, it doesn't block the actual active site, it binds somewhere else. And when it does that, it changes the shape of the enzyme, it changes the active site, right? So now the substrate can't fit in. It didn't block it, it just changed the shape. And now that it changed the shape, the substrate won't fit, and again, the enzyme can't do its job. Pretty simple stuff, right? And the next slide I'm going to show you shows you both a substrate imposter and this one that binds at a remote site, all in the same picture. Here's an enzyme working normally up top, right? Substrate fits in the active site, good, it does its job. Then we have the two inhibitors. We have the enzyme inhibition by a substrate imposter. So again, this thing, it's not the exact shape as the substrate, but it's the right shape where it can fit in the active site. So because it fits in the active site, the substrate can't fit in the active site, and the enzyme can't do its job. And of course, the other one here, this is the inhibition of an enzyme by a molecule that causes the active site to change shape. So an inhibitor goes somewhere else, that's not the active site, it's just somewhere else on the enzyme, but that causes the active site to change shape, therefore the substrate can't fit in, and again, the enzyme cannot do its job. Pretty simple stuff, right? Not much else to say about that. Any questions so far? All right. The book points out that the binding of the inhibitor can be reversible. As a matter of fact, it almost is. Yes, good question.
Okay, can you go back?
Yeah.
So the inhibitor,
Mhm.
why doesn't it go in like after the substrate? Because like it's kind of before.
This one?
Why wouldn't it go in after? So then obviously then they both fit. Right?
You mean go in after, like here?
Yeah.
Well, if I'm understanding correctly, it just won't fit there. It only fits here.
No, I mean like so the substrate and the top one it goes in there, so it's fine.
Uh-huh.
So why wouldn't the substrate go in first?
Oh, here?
Yeah, and then
It would. Actually, great question. So she's saying, why doesn't it just go in like right here? And it would, right? Without the enzyme inhibitor in there, it would fit just perfectly. So this picture, kind of imagine like this is just one situation. The substrate's not even around, right? And then it goes in, and then the substrate shows up. So you're right. If the substrate were to be around at that moment before the inhibitor went in, it would fit. But as soon as that inhibitor goes in and it's changed its shape like it has here, it won't fit. Good question. Any other questions? All right, so again, like I said, the binding inhibitors can be reversible. I think a better way of saying that is that very often it is reversible because again, your cells need a way to say, all right, I need you to make this, or then, okay, just stop putting resources into that, you made enough of them. Because again, more products are produced than are needed sometimes. Yeah, it's basically everything I said earlier. Product reversibly inhibits the enzyme, it keeps the cell from wasting resources. So basically the only thing new on this slide, the only thing I'm saying new on this slide, is that sometimes the product of the reaction itself can act as an inhibitor. So I'm not going to get that deep on anything. But that would make sense though. Again, your cells don't want to waste resources, energy and resources, building stuff that doesn't need to be built. So if you've got a lot of them floating around in your cell, a lot of the product, sometimes those products will hook back up to the enzyme and keep it from working. Which makes sense. Like, hey, there's enough of us, there's a high concentration, therefore you can stop working and stop building these things now. Anyway, your book gives some examples of this. Many drugs work by inhibiting enzymes. Penicillin, for example, blocks the active site for bacteria that the bacteria uses to make cell walls. So basically, in a way, you might say that penicillin doesn't kill the bacteria, but it keeps the bacteria from making that protective cell wall. And then without the protective cell wall, other things can kill the bacteria. Ibuprofen, it works by inhibiting an enzyme that sends pain signals. So when when you're in pain, that spot is sending signals to your brain and says, hey, I'm in pain. So when you take ibuprofen, it's not like the pain goes away, it's just that communication slows down or stops. And of course, many cancer drugs inhibit enzymes that promote cell division. That'll be a big one later in the semester. We're going to talk about exactly what cancer is. And yeah, so when we get to that, keep in mind that that's how some of the cancer drugs work. And your book also points out that many toxins and poisons also work as inhibitors. For example, that first picture I showed you for chapter 5 with that dude in the yellow hazmat suit, I said, can we guess what that might be? And I told you that there was nerve gas. He was protecting himself from nerve gas, and he would learn how nerve gas works a little bit. So that's the point right there. Now you know how nerve gas works a little bit. You know that it works as an inhibitor. If you want to look that up, though, how does nerve gas work? What enzymes does it inhibit? If you're curious, how we get it decent independent work. Put a big X through this, though. Not because it's not true, but because I'm not going to be asking you any questions about that on the exam. Might be some on the study guide, I don't remember, but I won't ask you about these drugs on the exam. So any questions about enzymes? You're done with your introduction to enzymes. Now for the rest of the semester, at least the first half, we're going to be talking a lot about different types of enzymes, but this is all the introduction that we're going to do. Which brings us to what you need to know for lab today. It starts at noon, don't be late. Membrane function. You're going to learn about the membrane function. So first of all, cells regulate the flow of materials to and from the environment. We know that. That is one of the properties of life, right? Things that are alive can regulate their environment, their internal environment. So in the case of a cell, their internal environment is what's inside the cell. So the ability to do that is one of the properties of life. How does it do that? Well, if we did chapter 4, you would already be familiar with this word, but we skipped it. So I'm going to introduce you to it now. It's something called a plasma membrane. It's basically just the outside of a cell. I don't want to say cell wall because there's a difference between a cell membrane and a cell wall. We'll talk about that later. But yeah, a cell membrane is also called a plasma membrane. I'm going to put it next to it because I'm not going to ask you any of these questions about it, but we'll talk about it. As you can see here, it's a double layer of fat. So there's layer 1, there's layer 2, right? Layer 1, layer 2. Each one of those little balls is it's a lipid, right? It's a fat. And that's why it's called a phospholipid bilayer, because it's got 2 layers of fat. It also has, and this is the slightly more important part as far as this discussion we're having today is concerned, it has embedded proteins. The next slide I'm going to show you shows a bunch of these embedded proteins, but here's one right here. Here's another one, and we'll talk about the different types here in a second. Before I show you that next slide, does anybody have any questions about this? All right. As far as the exam is concerned, this is an important slide. And as far as, this is like the explanation of why we're talking about it. Like, oh, what do we care about this? Again, cells regulate the flow of materials to and from the environment. They can control their environment. That's one of the properties of life, and that's why this is important. So, here's a bunch of different examples. You do not need to know any of these examples, but we'll talk about them really quickly. Again, we said that there's proteins embedded in the phospholipid bilayer. So here we have 2 cells, right? Here's one at the bottom, here's one at the top, and we have a bunch of different examples. Here we have a protein that attaches the cytoskeleton, which is like the inside. Again, if we did chapter 4, you would know that. It attaches to the cytoskeleton, the inside thing that gives it its shape, so to speak. Let's see, what's this one doing? Cell signaling. So sometimes cells need to talk to each other. I think in chapter 1, you may have read something about information flow, and they used the pancreas as an example. We'll talk about insulin. So there's signals that tells the pancreas to make insulin. And, that's how that works. We won't get into it. We won't ever talk about this, as a matter of fact, for the rest of the semester. Enzymatic activity. Basically, it's just a regular old enzyme, except it's embedded into the phospholipid bilayer. So everything you just learned about enzymes, same thing, except in this case, they're not just floating around in your cytoplasm on the inside of your cell. They're literally embedded into the cell membrane. Here we have intercellular joining. So as you might imagine, your cells are attached, right? A lot of your cells are attached to each other. Otherwise, you just be a big blob of nothing. So how do they do that? Well, one of the ways they do that is through these proteins that are embedded in the cell membrane, and they join together. Cell-to-cell recognition. This is really big when it comes to stuff like the immune system, right? So your immune system will recognize cells that shouldn't be in your body and attack them, in theory, right? Or another way of saying it is your immune system will recognize cells that should be in your body and they don't attack them. How do they do that? Cell-to-cell recognition. What's the other one? So we're talking about, here we go. I'll save the best for last. It's not the best, but it's the most relevant because this is the one we're going to talk about in this chapter. Transport. So there are proteins, again, that allow things in and out. Because remember, like I said in this last slide, cells regulate the flow of materials to and from the environment. Well, that's one of the big ways they do it with these transport proteins. So that's what we're going to talk about for the rest of this chapter, is things coming in and out of the cell. So again, like I just said, an important function of membrane proteins is the regulation of transport in and out of cell, right? Your cells need to control the concentration of the molecules in them. Maybe it needs a lot of glucose, maybe it needs a little bit less glucose, maybe it needs sodium, maybe it needs potassium. And your cells can regulate that. If you download the PowerPoint, you can watch this little animation. This shows membrane selectivity. I'm not going to put you through it right now. Same thing here. Here's another animation that shows membrane transport. All right. Now for the stuff that you're going to need to know for the lab. Passive transport is the first thing we're talking about. And before we do that, your book doesn't say this, I don't think, but your lab manual did. What I'm about to talk about right now is called Brownian motion. Molecules vibrate and wander randomly. We've already mentioned this earlier. I think we were talking about, how water regulates temperature. It's way back in chapter 2. And I told you that molecules move around a lot, right? The more they move, the hotter things are. So this is coming back to that idea. Molecules move around a lot. And here's a good one. Here's another water molecule. If you were looking at one single water molecule, it's not like it just sits there in space. If you could somehow zoom in and see it, it does all kinds of stuff. In addition to just literally moving around, in a three-dimensional way, that thing itself, even if it's stationary, is also moving in different ways. Like so, try to picture this thing doing this, right? It's stretching like that. And this time, it's stretching alternately. And this thing is bending and flapping like a bird's wings, right? You don't need to memorize any of those. Those are just examples. The point is, molecules don't sit still. They're always moving, not only three-dimensionally, but as they sit there, they do all kinds of different things. So what happens? Well, they hit each other, right? So if this thing's stretching like that, and then it accidentally bangs that one, the thing's going to go flying off, and then it hits this one that's flapping around, and it goes flying off, right? So the more they move, the more things move around. That's where we get diffusion, as you already know. Because again, what I just described is Brownian motion. Diffusion is the movement of molecules from high concentration to low concentration. I always think this is easy and people are going to get it because this is, in my opinion, one of the most simple concepts of the semester, but people still mess it up. Diffusion is the movement of molecules from a high concentration to a low concentration until equilibrium is reached. So if I were to light a scented candle right now, it would be really like a very heavy scent, right? And probably the first person to smell it would be you guys. And then you guys. And then eventually Brock would smell it. Right? I don't know how long. Eventually, the scent would go throughout the whole room, right? But when I first lit it, there would be a difference, right? If you were to walk in the back door, you wouldn't smell it. And as you walk closer, it will be a heavier and heavier scent. Does that make sense? But once it's been burning long enough, then no matter where you go in the room, it's going to smell the same. And that is what equilibrium would be, right? It's when all those molecules, whatever it is that's giving that scent, just equal throughout the room. So no matter where you go, it's all going to smell the same. Yeah, so this next slide is kind of visualize it. You can see a membrane that's separating pure water from a mixture of dye dissolved in water. So imagine again, you put a drop of a dye on this side of the water. Eventually, those molecules are going to move around, right? They're going to bounce around, and as they're bouncing around, eventually they're going to get through that hole, and they're going to keep doing that until it's an equal number, right? Until equilibrium is reached. So again, you went from an area of high concentration to an area of low concentration until equilibrium is reached. And I should point out too that even though equilibrium is reached, the molecules are still bouncing around. They're still going from side to side, but the overall concentration is not the same, because that's what equilibrium is. The concentration is the same no matter where you're looking at. So any questions about this? I should point this out too, sorry. As it's diffusing, right, from this area of high concentration to low concentration, like I said earlier, this is all random. So even though it is going from high to low, still some of them are going to go back the other way. But overall, the net movement is going to be from this side to this side until it's equal. But that's just details. The most important thing here that you know is that we're going from high concentration to low concentration. This next slide I'm going to show you shows the same thing, except it's two different colored dyes on two different sides. So here we go. We know these purple ones are going to diffuse to the right. The red ones are going to diffuse to the left. And that's what happens until equilibrium is reached. And now you have, equal concentrations of both on both sides. Any questions about that? Someone will miss that test question. I don't know how, but remember, the key here is high concentration to low concentration. That is diffusion. Specifically, that is something we call passive transport. That is the diffusion of a substance across the membrane without the input of energy. So let me try to visualize there. I can write it there. Yeah. So we have this term diffusion that you've learned. And a subcategory of diffusion is passive transport. So passive transport is a type of diffusion. Make sure that's clear. So diffusion is not a type of passive transport. Passive transport is a type of diffusion, right? Because like I said, if I light a scented candle, and then that scent eventually makes its way throughout the room, that would be diffusion. But what it would not be passive transport, because we're not talking about the membrane. Specifically, in this class, of course, we were talking about cell membrane. So if we're talking about diffusion, talking about passive transport, and it's specifically the example of diffusion, but across the cell membrane. And again, because it's passive, it does not require an input of energy. Without input of energy, because it's passive. And again, here's another way of saying what I said earlier that you really need to know. If we're talking about diffusion, or in this case, a specific type of diffusion, which is passive transport, it is going down the concentration gradient. So there will be a question about passive transport. Those are the things you need to know about it. First of all, it's a type of diffusion. Second of all, it's specifically the case of diffusion, but across the cell membrane. Third, because it's passive, it does not require an input of energy. And then fourth, it's going down the concentration gradient. Now later, I'm going to tell you about active transport. If you're a thinker, hopefully you could probably already figure this out. It's probably a lot like the opposite of passive transport. Active would be the opposite of passive. So instead of not needing an input of energy, it would need an input of energy. Instead of going down the concentration gradient, it'll go up. But we'll talk about that when we get there. Any questions about what diffusion is or passive transport? All right. If you download the PowerPoint, you can watch that little video that shows diffusion in action. Again, I'm not going to take the time doing that. So, some things do not cross spontaneously through the cell membrane. So again, there's the cell membrane. This picture right here, though it says diffusion, this is also passive transport. Specifically, yes, it's diffusion, but that is passive transport, PT. But some things cannot get through the cell membrane. So even though there's an area, there's high concentration here and low concentration there, and nature just wants them naturally to move that way, down the concentration gradient, for some reason, some molecules can't do it. Like, for example, the phospholipid bilayer is hydrophobic. So maybe these things are hydrophilic. So because of that, they don't mix. So because of that, they cannot just go straight through the membrane. And because of that, they need a little doorway, so to speak. And that is what is called facilitated diffusion. So you need to know that too. Facilitated diffusion is a type of passive transport, right? It doesn't require the cell to expend energy, but it provides this little pathway, a little doorway. So if we crank the temperature up in here to 120 degrees, and that air molecules are moving around really fast, which way do you think they would go? Do you think more air molecules would come in here, or do you think air molecules would leave like under the crack of the door? If they're really moving fast in here, and they're really slow out there. That's a tough one. All right, let's go back to the idea of the scented candle. So if I had a really, really big scented candle in here, it's smelling a lot, but not out there. You think fresh air is going to come in here, or do you think the scented air is going to go that way, out to the hallway? Into the hallway, right? Because that would be diffusion. It's an area of high concentration that's sent in here to an area of no concentration out there. And it would, right? It would probably go right under the crack of that door. Because it can, it's just air molecules. But imagine if we filled this room with 500 people, and there's 0 people out in the hallway. What's naturally going to happen? People are going to want to go from this area of high concentration to the area of low concentration, right? But they can't because the door's shut, right? So yes, nature wants them to go from here out to there, but the door's shut and won't let them. So what would you do in that case? Don't overthink it. Open the door, right? You just open the door. So yes, nature wants them to go from high concentration to low concentration, but they can't because the door's in the way. That's what facilitated diffusion is. It gives you that protein right here. It just opens the door, so to speak. It gives them a way to actually get through. The next word for attendance is going to be cell. Like I'm underlining. Anyway, any questions about what facilitated diffusion is? So to be clear, again, passive transport is a type of diffusion because we're specifically talking about through a membrane. And then another type of passive transport is the facilitated diffusion. All right, there's no questions about that. Let's talk about the other thing you need to know for the exam. For the exam too, but for lab. Osmosis. Osmosis and water balance. Osmosis, and you need to know this, is the diffusion of water across a selectively permeable membrane. So now we're getting even more specific. We're still talking about diffusion. This is still a subcategory of diffusion, except now we're being really specific. It's not just the diffusion of any old thing. It's specifically the diffusion of water. And it's across a selectively permeable membrane. As far as we're concerned, you might as well just say, not for the exam, because you need to know that wording for the exam, but you could just say across a cell membrane, right? That's what we're talking about. When we say selectively permeable, we're talking about a cell. Yes, there's other ways to do it that we don't get into, but this is a biology class, and we're talking about the cell membrane. And then there's words that we've already been introduced to, solute, solvent, and solution. Right? The solute is the substance that's dissolved into a solvent, and that resulting mixture is called a solution. So if I put salt into water and get salt water, the salt would be the solute, the water that I put it into would be the solvent, and the resulting salt water would be the solution. Any questions about that? And I'll go through that pretty quickly because, again, you've already been familiarized and introduced to those words. If you download the PowerPoint, you can watch this animation of osmosis. It actually gets a little bit more complicated, and we don't have enough time to get into it. So when we come back on Friday, we'll talk about how osmosis works. Obviously, it's water going across the membrane, but what happens? How does it, what dictates it? All right. Does anyone want to volunteer to take the picture? All right. We'll take it, whoever wants to be in the WBSU picture, we'll take it up on the second floor, but that way a little bit. You'll see. Let me close out all this and I'll show you. Yeah.